Greetings from Everyday GMP. I am your presenter from Everyday GMP. This is series of video. We will discuss all the definitions and term used in EU guidelines for good manufacturing practice for medicinal products for human and veterinary use. In this video we will cover terms starts from alphabet P, Q, R, S and T. Further series will be covered in other videos. Total 6 videos are in this series. This is fifth and second last video. Links of previous videos are in description. Keep watching all videos, so let's start. 1, 2, 3. Let's start. We will learn today so many terms and definitions like what is passed through hatch, the parasite, the patient. It could be human or animal post-aseptic processing, terminal heat treatment, what are pyrogens, what is restricted access barrier system, RABS, sterile grade raw material rapid transfer system, port, RTP. The single-use systems, SUS, sterilized grade filter like 0.2 micron pore size filters in the last turbulent airflow. Let's move to main session. EU GMP Guidelines Annex 1 series of definitions, video number 5. Term starts with alphabet P, Q, R, S and T. In the world of pharmaceuticals, precision and adherence to regulations are paramount. Understanding the key definitions and terminology laid out in the European Union Good Manufacturing Practice Guidelines, Annex 1, is not just important, it's essential. Join us on a journey through the intricacies of pharmaceutical manufacturing as we dive deep into the terminology that shapes this industry. In this comprehensive video series, we will demystify the definitions used in the EU GMP Guidelines Annex 1, breaking down complex concepts into easily digestible explanations. Exploring pharmaceutical definitions from EU GMP Guidelines Annex 1, unlock the language of pharmaceuticals and dive into the core terminology that shapes the industry. Join us on this educational journey to understand the key definitions that ensure precision and quality in pharmaceutical manufacturing. Subscribe and let's begin. So before start, subscribe the channel and press the bell icon. So let's start. Parasite, the tube of polymer extruded by the BFS machine from which containers are formed. Blow molding, or molding, is a manufacturing process for forming hollow plastic parts. It is also used for forming glass bottles or other hollow shapes. In general, there are 3 main types of blow molding: extrusion blow molding, injection blow molding, and injection stretch blow molding. The blow molding process begins with softening plastic by heating a preform or parasite. The parasite is a tube-like piece of plastic with a hole in one end through which compressed air can enter. The patient, human or animal including participants in a clinical trial. These are subjected for clinical trials. Pass through hatch, synonymous with airlock. See airlock definition, but typically smaller in size. Post-aseptic processing, terminal heat treatment, a terminal moist heat process employed after aseptic processing which has been demonstrated to provide a sterility assurance level, SAL, is less than or equal to 10⁶, but where the requirements of steam sterilization, for example, F0 is greater than or equal to 8 minutes, are not fulfilled. This may also be beneficial in the destruction of viruses that may not be removed through filtration. Pyrogen, a substance that induces a febrile reaction in patients receiving injections. Pyrogenicity presents a challenge to clinicians, medical device manufacturers, and regulators. A pyrogen is, by definition, a substance that produces a rise in temperature in a human or animal. Pyrogens are differentiated into exogenous and endogenous pyrogens. Rapid Transfer System, POT, RTP, a system used for the transfer of items into RABS or isolators that minimizes the risk to the critical zone. An example would be a rapid transfer container with an alpha, beta port. Raw material, any ingredient intended for use in the manufacture of a sterile product, including those that may not appear in the final drug product. Restricted Access Barrier System, RABS, system that provides an enclosed, but not fully sealed, environment meeting defined air quality conditions, for aseptic processing grade A, and using a rigid wall enclosure and integrated gloves to separate its interior from the surrounding cleanroom environment. Continue. Restricted Access Barrier System, RABS, continue. The inner surfaces of the RABS are re-disinfected and decontaminated with a sporicidal agent. Operators use gloves, half suits, RTPs and other integrated transfer ports to perform manipulations or convey materials to the interior of the RABS. Depending on the design, doors are rarely opened, and only under strictly predefined conditions. Single-use systems, SUS, systems in which product contact components are used only once to replace reusable equipment such as stainless steel transfer lines or bulk containers. Continue. SUS covered in this document are those that are used in manufacturing processes of sterile products and are typically made up of disposable components such as bags, filters, tubing, connectors, storage bottles and sensors. Sporicidal agent, an agent that destroys bacterial and fungal spores when used in sufficient concentration for specified contact time. It is expected to kill all vegetative microorganisms. Examples include phenols, chrysols, chlorhexidine, quaternary ammonium compounds and sublethal concentrations of chlorine-releasing agents and glutaldehyde. Sterile product. For purpose of this guidance, sterile product refers to one or more of the sterilized elements exposed to aseptic conditions and ultimately making up the sterile active substance or finished sterile product. These elements include the containers, closures, and components of the finished drug product, or a product that is rendered sterile by a terminal sterilization process. Sterilizing grade filter, a filter that, when appropriately validated, will remove a defined microbial challenge from a fluid or gas producing a sterile effluent. Usually, such filters have a pore size equal or less than 0.22 micrometers. Terminal sterilization, the application of a lethal sterilizing agent or conditions to a product in its final container to achieve a predetermined sterility assurance level, SAL, of 10 to the power of 6 or better, e.g. the theoretical probability of there being a single viable microorganism present on or in a sterilized unit is equal to or less than 1 × 10⁶, 1 in a million. Turbulent airflow, air that is not unidirectional. Turbulent air in clean rooms should flush the clean room via mixed flow dilution and ensure maintenance of acceptable air quality. That's all for today's video on terms and definitions used in pharmaceuticals. If you found this information helpful, give us a thumbs up, subscribe to our channel, and hit the notification bell to stay. If you have any questions or want to share your journey, leave a comment below. Until next time, Keep dreaming big and reaching for your goals.